Welcome back to the Deep Dive. And I have to say, a huge, massive welcome to our very first worm podcast, whole organism analytics podcast, deep dive of the year.
It is genuinely great to be back.
Today is Tuesday, January 20th, 2026. And a very happy new year to you. I really hope you managed to disconnect, recharge, maybe eat way too much food over the holiday break.
Oh, definitely.
I have a feeling 2026 is going to be a massive year for biological discovery, and honestly, I've been itching to get back behind the mic.
Same here. Happy New Year to everyone listening. And we are kicking off the year with a topic that, on the surface, sounds a bit like the plot of a true crime documentary.
It really does. The working title for today is Partners in Crime. But we aren't talking about, Bonnie and Clyde or some high-stakes bank robbery.
No, we're digging into the dirt. Literally to look at a conspiracy happening right beneath our feet.
Right there in the rhizosphere.
Yeah.
And specifically, we're unpacking a fascinating new review paper.
That's right. It's titled Partners in Crime, Elucidating the Molecular Underpinnings of Nematode Pathogen Disease Complexes.
And that was just published in MPMI, that's Molecular Plant Microbe Interactions, by Alison Blundell and her team. Brand new for 2026.
Right. Now, to set the stage, we usually talk about plant-parasitic nematodes, PPNs, as the solo villains. And for good reason. On their own, they are an absolute wrecking ball for global agriculture.
The numbers are staggering.
They are. The estimates put the damage at around $173 billion annually.
For a microscopic worm. It's just incredible.
It is. But here's the hook for today's deep dive. The real danger, the scenario that keeps plant pathologists up at night, isn't the nematode acting alone.
It's when it teams up.
It's when it teams up with other pathogens, bacteria, fungi, viruses. When they coordinate, they create what we call disease complexes.
So the mission for this deep dive is to understand how they do it. How do these tiny worms conspire with other microbes to take down a plant?
And crucially, why the result is often so much worse than just adding the two diseases together.
Okay, let's get into the setup.
Yeah.
You use the term disease complex. I feel like we need to be precise here, because that could mean a few things. Is this just bad luck?
Right.
Like a plant gets a fungal infection, it just happens to have nematodes, two bad things at once.
Not exactly. That would just be a co-infection. A disease complex implies synergy.
Synergy. Okay, that's a buzzword we hear a lot, but what does it mean in the soil?
It means the interaction creates symptoms that are way more severe than if you just added the damage of pathogen A to pathogen B. It's not 1 + 1 = 2.
It's 1 + 1 = like 5 or 10.
Exactly. It's the difference between yield loss and total crop failure.
Okay, that puts the stakes in perspective. So who are the main characters in this drama?
Well, obviously, our antagonists are the nematodes, and we really need to distinguish between two main lifestyles here because they have very different, let's say, modus operandi.
Right. We're sticking to the crime metaphor. You've got the ones that squat and the ones that are more like hit-and-run artists.
That's a great way to put it. You have the sedentary endoparasites. These are the heavy hitters like root knot nematodes and cyst nematodes. They get inside a root, find a spot, and they do not leave.
The squatters. Got it.
Then you have the migratory ones like root lesion nematodes or dagger nematodes. They are the hit-and-run artists. They move through the soil and the root, grazing, slashing, moving on.
And their accomplices?
The partners in crime span the whole microbial world. We're talking about really nasty bacteria like Ralstonia, fungi like Fusarium, and a whole host of viruses.
So have we known about these gangs for a while? This is a new discovery.
Oh, we've known about the phenomenon for ages. The paper points out reports going all the way back to 1892. A researcher named Atkinson.
Wow, 1892.
Yeah, but for over a century, we just knew that it happened. We didn't really know why. What makes this 2026 paper so exciting is we're finally cracking open that black box and looking at the molecular mechanics of the partnership.
So let's look at the crime scene. If we break this down like a heist, the first step is always getting in the door.
Exactly. Breaking and entering. And this brings us to the simplest, most basic form of cooperation, wounding.
The physical breach.
Right. All plant parasitic nematodes have a stylet. You should think of it as a hardened hypodermic needle for a mouth. They thrust it into plant cells to feed.
And that's going to leave a hole.
It creates a physical opening. Now, for some aggressive pathogens, that doesn't really matter. They can break in on their own. But think about the weaker ones, the opportunists.
The ones that can't get through the door by themselves.
Exactly. For them, the nematode just kick the door in. But the paper highlights something else, which is really interesting. It's not just about the hole, it's about what comes out of the hole.
Leakage.
Leakage. When a nematode damages a root, all the cellular contents spill out. Sugars, amino acids,
All the good stuff.
the dinner bell. This triggers a process called chemotaxis. You have to remember, soil bacteria aren't just wandering randomly. They can sense chemical trails. They follow this plume of nutrients right to the source.
So it's not just an open door. It's an open door with the smell of fresh pizza wafting out. The nematode bites, the root bleeds nutrients, and the bacteria just swarm to it.
That is the perfect analogy. And the migratory nematodes, like Proliferation, the root lesion nematodes, they're particularly messy eaters. They literally tunnel through the root tissue, leaving a trail of dead, leaking cells.
Which I assume just invites rot.
It does. It causes massive root rot because every microbe in the neighborhood is drawn to that scent trail.
Okay, so strategy 1 is smash and grab. Make a mess, let the opportunists in. But you mentioned the sedentary nematodes, the squatters. They're more sophisticated than just breaking windows, right?
Much more sophisticated. And this brings us to the second mechanism, the safe house.
The safe house, I like it. Explain that.
So sedentary nematodes, like root knot nematodes, they don't just eat the plant, they fundamentally remodel it. They hijack the plant's own development to build these specialized feeding structures.
These are the giant cells or syncytia I've read about. They're like supersized, nutrient-rich cells.
Right. Yes, they are metabolic sinks. The plant is tricked into pumping sugars and nutrients into them, basically force-feeding the nematode. It's a 5-star luxury hotel built inside the root.
But I see the risk here. If you build a mansion in a bad neighborhood.
Exactly. That nutrient-dense hotel becomes a prime target for secondary pathogens. The paper highlights the bacterial wilt pathogen, Ralstonia. It's been found specifically colonizing the galls that root knot nematodes create.
So the nematode does all the hard work of concentrating the food, and the bacteria just moves in and takes over.
It hijacks the hijacker. And the same thing happens with fungi, like in the Fusarium wilt complex. Fusarium exploits this rich, modified root, and here's a fascinating twist.
Ooh, I love a twist.
It's not always a happy partnership. We call them partners, but sometimes they're more like rivals.
How so?
Fusarium is what we call a necrotroph. It feeds on dead or dying tissue. It actively kills the root cells to eat the debris.
Okay.
But the root knot nematode is an obligate biotroph. It needs living tissue to survive. It needs that giant cell to be alive and pumping out food.
I see the problem. If the fungus moves in and kills all the cells, the nematode starves.
Precisely. So while the plant dies much, much faster, which is that synergistic effect we talked about, the nematode's own reproduction often goes down.
That is wild. It's like a robber teaming up with an arsonist. The robber picks the lock, but the arsonist burns the bank down before he can get the money out.
That is a shockingly good summary. The plant is the bank, and it gets destroyed, but the robber nematode often loses out in the end.
Okay, so we have breaking and entering, and we have the safe house. But there's a third role for the nematode, right? The smuggler.
The smuggler. This is where we talk about nematodes acting as vectors, specifically for viruses.
So the nematode is the taxi.
A taxi, or maybe a better analogy, is a dirty needle. This is a highly specific relationship. It mainly involves certain nematode families, the Longidoridae, those are dagger nematodes, and Trichodoridae.
When you say specific, does that mean any dagger nematode can carry any virus?
No, and this is where the molecular science gets so cool. The virus doesn't replicate inside the nematode. The worm isn't infected.
So it just rides along.
It sticks. As the nematode feeds on an infected plant, viral particles get sucked up. They adhere specifically to the lining of the nematode's stylet, or esophagus, a part called the odontophore.
It just sticks to its throat.
It binds there, but it's a molecular lock-and-key mechanism. The virus has a specific coat protein. The paper mentions one called 2-CCP that fits perfectly into the receptors on that nematode's throat lining.
So if you change the coat protein,
the nematode can't carry it, it won't stick, it would just pass through the gut and be excreted. If you swap that coat protein, the virus becomes non-transmissible by that nematode.
That is incredible specificity. It's like a custom-fitted holster.
It is. And it's not just viruses inside. The source also mentions foracy, which is basically hitchhiking on the outside of the nematode.
Like burrs sticking to your socks when you walk through a field.
Exactly like that. Bacteria can stick to the cuticle, the nematode's skin. The paper gives the example of Rhethibacter bacteria sticking to the surface of Anguina seed gall nematodes. The nematode just provides the transportation.
So we have physical entry, creating a buffet, and smuggling. But there's one more layer to this, and I think it's the most sophisticated one. It's about hacking the plant's alarm system.
This is really the frontier of our understanding, immune suppression.
Right, because a plant isn't defenseless. It has an immune system.
It absolutely does. The first line of defense is called PTI, or pattern-triggered immunity.
Can we break that down? How does PTI work?
Imagine the plant cells have little radar dishes on their surface. They are constantly scanning for patterns associated with microbes, like flagellinin, which is a protein in bacterial tails.
So the plant sees a bacteria and says, hey, that's not supposed to be here, and it pulls the fire alarm?
Basically, yeah. It triggers chemical warfare, a burst of reactive oxygen species, and it reinforces the cell walls with callus to lock the pathogen out. But nematodes have evolved to shut this all down.
How do they do that?
They secrete proteins called effectors. These are their hacking tools. The paper gives a great example, an effector called MimSP32 from the root knot nematode.
Catchy name. What does it do?
It interferes with the production of jasmonate. Jasmonate is a key defense hormone. It's the signal that rallies the troops. The nematode effector essentially cuts the phone lines so the message can't get out.
Wow. So the nematode cuts the phone line.
It cuts the lines and it drugs the guard. Another effector they mention, MinTCTP, binds calcium inside the cell. Calcium is a critical signal for danger. By locking it up, the nematode stops the alarm from ever ringing.
And here's the link to the disease complex. By dampening the immune response to save itself, the nematode inadvertently disables the alarm for everyone, right?
That is the key. The plant becomes immunosuppressed. Secondary pathogens, bacteria, and fungi that might have been stopped at the door can now just waltz right in.
But wait, don't plants have a backup system, like a SWAT team if the regular guards fail?
They do. It's called ET effector triggered immunity. The plant has internal sensors called NLR receptors that can detect if someone is messing with the system inside the cell.
And I'm guessing the nematodes hack the SWAT team too.
They do. It's an evolutionary arms race. The paper describes an effector called RHA1B in potato cyst nematodes. It acts as a ubiquitin ligase.
Whoa, okay, ubiquitin ligase. Let's find an analogy for that one.
Think of it as a label maker. It goes around tagging the plant's own defense proteins with a trash me sticker.
No way.
Yes. The plant's own waste disposal system then sees the tag and destroys its own defense machinery.
That is unbelievably devious. It tricks the plant into disarming itself.
And it gets worse. This specific effector suppresses something called the hypersensitive response.
The hypersensitive response. That's when the infected plant cell commits suicide to stop the infection from spreading, right?
Correct. It's a scorched earth tactic. If I die, the infection dies with me. But the nematode effector stops the cell from pushing the self-destruct button because it needs that cell alive to feed.
But by keeping the cell alive.
It keeps the door open for the fungus or bacteria to keep spreading. If the plant can't trigger that localized cell death, the secondary pathogen can just run wild.
So let's bring this all back to reality. What does this molecular hacking actually mean for a farmer growing tomatoes or potatoes?
It's a management nightmare. That's the bottom line. Because normally you rely on resistant plant varieties, right?
Yeah.
You buy the tomato seeds with the little codes on the packet.
Right, VFN for Vericillium, Fusarium, nematodes.
Exactly. Take the My1.2 gene in tomato. It's a famous resistance gene. But here's the problem. That resistance might stop the nematode from feeding successfully, but it doesn't always stop the initial penetration.
So the nematode still punches the hole.
It still punches the hole, and even if the nematode dies moments later because of the resistance gene, that initial wound or that momentary suppression of immunity is enough to let something like Ralstonia bacterial wilt get in.
So the resistance gene works against the nematode, but the plant still dies from the bacteria.
Exactly. The resistance is effectively broken by the complex. The source calls this predisposition. The nematode predisposes the plant to attack.
Does the timing matter here, or is it just bad news whenever they show up together?
Well, timing is a huge factor, according to the research. The disease is most severe when the nematode arrives before the bacteria, specifically about 10 days prior.
10 days. Why that specific window?
That seems to be the sweet spot. It gives the nematode enough time to establish its feeding site, suppress the local immunity, and really roll out the welcome mat. If the bacteria gets there first, the effect is much less devastating.
So from a farming perspective, if you have nematodes in your soil, you're sitting on a powder keg, even if the nematode numbers aren't technically that high.
Yes. You might look at a soil test and think, oh, my nematode count is below the economic threshold, I'm fine. But if Fusarium is in that soil too, you could lose the whole crop. The threshold for damage just plummets.
It completely changes how you have to think about pest control. You can't just treat for one thing in isolation.
We have to manage the whole system.
It's a lot to take in. Just thinking about the coordination going on down there in the dark.
It really is an ecosystem of crime.
So, to quickly summarize what we've learned from this Blundell paper, nematodes are not just solo actors. They are the ringleaders of the soil underworld.
They break in with their stylets, creating runes and leaking nutrients that act like a dinner bell for other pathogens.
They act as smugglers, vectoring viruses like dirty needles or giving bacteria a ride on their backs.
They build safe houses, those nutrient-rich giant cells that bacteria and fungi then invade and exploit.
And maybe most terrifyingly, they hack the plant's alarm system, suppressing its immunity so that everyone can feast.
That covers the main mechanisms perfectly.
Before we wrap up, I want to leave our listeners with one last thought. We spent this whole time talking about how bad this all is, but is there a flip side? Can we use this knowledge?
There's a really provocative question raised at the very end of the paper. We talked about 4Z microbes sticking to the outside of nematodes.
Right, the hitchhiker.
Well, the authors ask, could we engineer beneficial microbes to stick to nematodes?
Oh, that's interesting, like a biological Trojan horse.
Exactly. Imagine a biocontrol bacteria that's engineered to grab onto a nematode's cuticle. But instead of causing disease in the plant, it attacks the nematode itself.
So you turn the smuggler into a delivery driver for the good guys.
Essentially. Instead of the nematode delivering a virus, it delivers its own destruction or maybe a plant beneficial compound. It's science fiction right now, but it's the kind of thinking this molecular understanding allows.
That is a fascinating idea to end on, turning their own weapons against them.
It's the kind of innovation we need for 2026.
Absolutely. Well, thank you so much for walking us through this. It certainly makes you look at a patch of dirt a little differently.
My pleasure. It is a whole world down there.
And a huge thank you to everyone listening to this first deep dive of the year. We hope you enjoyed this look into the microscopic underworld. Here's to a year of discovery. We wish you all the very best for 2026. Thanks for listening to the Worm Podcast Deep Dive, and we'll catch you next time.